Copyright University of Auckland, all rights reserved.
So I was recently given the opportunity to talk at Nerd Night, which was just on Tuesday, but some people didn't manage to make it, and I thought it'd be cool for everyone who's actually voted to have the opportunity to hear what the project's all about like they did. I'd like to say a very big thank you. They were awesome. Bought me a couple of beers, and there were two other really great talks. One was on flash photography and the history of it, and the other one was a guy from Arkham City Comics in Royal Oak. Fantastic night all around. But I thought I'd re-record my talk now, so those that couldn't make it could actually see, and thank everyone hugely for all their support, promotion and voting. I'm currently still in the top 5, so hopefully we'll get some money to actually do this bat translocation work. Where did it all start for me? Well, it was on Rangitoto. I was a first year. I was incredibly annoying. I probably still am, but I was really annoying then, and I was voicing the opinion that plants were boring. And my demonstrator was very, very tolerant and encouraging, and did his job and inspired a young biologist by pointing at Acedia banksii, which you can see here in the top right-hand corner, and saying, well, look at this plant. It has all these cool plant-animal interactions. Right now, possums are spreading the seeds and doing a little bit of the pollinating from pollen on their fur between the plants. Bats normally do it, but they're about to get rid of the possums, so that's going to have a huge effect on the ecosystem and the restoration of the island and its natural succession. And what I heard was somebody should translocate bats. And we really, really should, because unlike possums that quite often eat the flowers, and as a result you don't get any pollination at all, or very, very little, bats have evolved alongside New Zealand's plants to be a pollinator. And in some cases, like with this New Zealand woodrose, Dactylanthus torii, they are the preferred pollinator. Dactylanthus is endangered as well. It was highly prized for its roots, because the plant would burrow in to the roots of surrounding trees as a parasite and extract out some of the nutrients from the roots of neighbouring plants, and would cause this beautiful rose-type scarring on the roots. So people used to come along and dig up the plants so they could carve these beautiful trinkets. Unfortunately, this caused the plant to become endangered. And also, the reduction of short-tailed bats throughout New Zealand in terms of their range rapidly shrinking and the populations going extinct didn't help the plant out very much either. So when we're talking about translocating bats, we're not just doing it for the bat's sake, we're doing it for the sake of the entire ecosystem, because they're a very important part of New Zealand forest. So throughout my undergraduate degree, I learned about translocations and that sort of thing, and I discovered that most people tend to focus on 2 areas. One is, what's the quality of the habitat, because crappy habitat equals likely to fail, and how many you're taking there. Low numbers also means you're likely to fail. There's always a few individuals that die during translocations, because animals aren't immortal. They die naturally every year anyway. So if you have too few and they die, you may not actually end up with a viable population. You have inbreeding problems, all those sorts of things. And there is no denying that these 2 variables are incredibly important for the success of a translocation. But they don't matter in any way, shape or form if the animal leaves the release site or the translocation site. And this happens a lot more than we care to admit. Sometimes it's dispersal, like with the North Island robin. Other times it's flat-out homing, like the shore plover. The time they tried to translocate that little guy, they bet the researchers back to the capture site. And it has been suggested as the reason why the first translocation attempted in New Zealand of the short-tailed bat failed. Globally, we do not know of any successful bat translocation to date. So what about that first translocation? Well, it happened in 1994, so well over 20 years ago. They only took 25 bats of each sex, and it was from Codfish Island, which is at the top west side of Stewart Island, across to the other due east to Ulva Island. And this translocation wasn't actually done specifically for the establishment of a new bat population. It was more of an insurance policy, because they were actually doing a poison drop on codfish, and DOC, being conservative, wanted to ensure that they didn't get any secondary poisoning of bats, so they took a whole lot into captivity, and then sort of as an afterthought, they went, hey, we'll just try doing a translocation. It could have worked. Maybe the bats wouldn't have left the site. They may not have wanted to fly across the water. You don't know until you try. And so you can see codfish there at the top left-hand side of the screen, and Ulva is a little island in Petson's Inlet at the bottom right. And so we've got 4 possibilities for what happened to the bats. Because as soon as it was a hard release translocation, this means you put the animal down and you just let it go. There's no extended holding of it or feeding of it. So they just put the bats in some trees. As soon as dusk came along, they let go of the entrance and the bats just disappeared within 15 minutes. And we've got 4 possibilities for what happened to the bats. First one is the bats flew all the way back straight to codfish and pool to plover. Or maybe they flew around looking and eventually they made it back to codfish after a lot of searching. They could have done that. They could have flown around and then eventually given up and just settled somewhere on Stewart Island. Or we could have had the worst possible case. The bats could have flown out to sea and drowned, and we don't know what happened to them. They were never heard or seen from again. As it turned out, just as a little side note, that poison drop went out around perfectly. There's no predators on codfish anymore, and they didn't find any evidence of any secondary poisoning. So we're going to ask the question is, why do animals navigate? This is kind of central to the entire thing. And it's kind of weird when you look at some of the places animals navigate from. You know, who would want to go and live on an ice floe? But the reason for it is because resources aren't uniformly distributed in space and time. So any organism that can go from one location to another accurately is going to outcompete anyone that has to do random search. As a result, navigation is a universal trait that we find across so many different species and is so beneficial, it probably evolved very, very early on, which means it's more likely to be the default ability rather than something special, which is quite good because you can develop an animal navigation model and then apply it to each species to see how it fits. And there's some problems with navigation and homing studies. First of all, when you're selecting your study animal, first of all, it's got to be physically able to home. So it's got to be able to get past the geographic barriers and that sort of thing. The home has to be well defined. There's no point in doing a homing study from a location the animal's got no intention of returning to. So you've got to be able to reliably identify it once it returns, so there can't be any trap avoidance. Being easily transported and that sort of thing is also handy, which is why we don't do very many homing studies on whales. All of these factors makes selecting the humble homing pigeon a really intelligent choice. It is the best thing for this sort of study and what most of our information about navigation comes from, the species. So given that that's the most intelligent choice, you've got to wonder about my brain cells for picking something that's almost the total opposite. Bats are nocturnal, they're cryptic, they move around, they're hard to catch, hard to track. In fact, the only thing that they've got similar is that they're easy to actually handle. They're not going to fight me too much. But aside from that, they're the worst thing you could work on if you wanted to study navigation. But I wanted to study bat navigation, so there was no choice. But I had choice between 2 species here in New Zealand. The short tail, which I've been talking about, and the long tail bat. And I ended up working on the long tail bat for my masters. This was because it's a very fast, strong flyer. It doesn't chew off the transmitters. It could be permanently banded and identified. It's part of the largest superfamily, so it's got a very generic ecology, would make the best model. Compare that to the short tail, which is pretty much the opposite of all of those things. And at the time of me doing my masters, there was no permanent form of identification. Now we pit tag them as a way of identifying individuals, but prior to that, we couldn't ban them because the bans would cause them aggravation. So for my masters, I had 3 main objectives. The first one is to determine if Grand Canyon Cave, this is one of the few places in New Zealand where bats are actually found in caves. The rest of the time, they're all in trees. Whether that was like a loft, whether they used that on a regular enough basis for us to use it as a home location. Because they noticed that the use of this cave peaked in November, trailed off to small numbers in January, February, but we didn't know if this was a continual turnover or a residence group of bats. Then, if it was a loft, determine if these bats could home. Then finally, if possible, qualify the release behaviour. See what sort of cues they're using to navigate. If we understand the cues, we can understand the navigation system. So my site was down in Peopea, which is just due west of Taupo, on a very large sheep farm. The cave was very, very long. It's about 400 metres from running east to west, 30 metres high, and anywhere up to 10 metres wide, although it can be quite much narrower than that. I was using radio telemetry, so I trapped my bats using harp traps and set up 2 sites, and one at each entrance of the cave, to monitor any radio telemetry signals that we'd get from the bats with computers to record. And then I put little radio tags on the bats. And you can see a little long tail here with the radio tag glued to its back. It only stays on the bat for about 2 weeks at the most, and then the bat just grooms it off, the hair grows through, and you can't tell the difference. We've caught bats that have been radio tagged about 5 to 6 years later, identified by their armband, and they're perfectly fine. So it's a really good method of actually tracking your animal around. And what I found is that we do actually have a resident population of bats. It's not a huge turnover running through every night, and they do return quite regularly. The one bat that we didn't make it back, we still suspect did, because one night we actually caught a bat without a tag on its back, put it in a bag for me to come and take a look at it, but by the time I got to the trap, it had wriggled its way out of the bag and flown off. So I'm pretty sure that bat 209 actually made it back, but we can't say for certain. But even without that, we have a high rate of reuse of that cave, close to 90%, which is good enough for our study. So the next stage was to take bats in increasing distances away from the cave, starting at 5 kilometres and then moving within their home range and moving finally to 20 kilometres well outside their home range, and let them go. And what we found was the bats all zoomed back quite quickly. Again, one of the bats wasn't detected again. This could potentially be due to transmitter failure. That does happen about 1 in 10 times. And because the bats that we did make it back were either back at the cave the following night, and a few of them that took 3 days or so to make it back. When they left the release site, I managed to follow them into the familiar home range. So it definitely wasn't the distance or knowing which direction it was. It's just that was towards the end of the year where they used the cave less regularly. So, how are they doing this? Clearly, they've got some sort of map, sort of like this cartoon is suggesting. And for that, we need to look into the literature and see what what navigation theory says. The main central dogma was put forward by a guy called Kramer. I checked, he wasn't the guy from Seinfeld, but he proposed that bats and birds and everything else does the same thing that humans do. Before they can determine which direction they need to head in, they need to know where they are. And this is called the map step, and the second bit is called the compass step. So you need some sort of landmark or cue that you can use as a compass to maintain a constant direction. And we know, through lots of other research, that birds and bats and everything else uses celestial orientation first, and then if it's like a cloudy night, they fall back onto magnetic compasses. So celestial is dominant over the magnetic. And finally, once you get within your familiar area, you go on to landmarks. Some people did some research using homing pigeons. They gave them frosted lenses, and these frosted lenses allowed them to see the sun, but not to really make out buildings. The pigeons managed to make it back within 5 kilometres, and then they just walk around bumping into things, not working out where they were, and you'd have to go catch them with a net. This is in the 1970s, so we don't get to do this sort of stuff anymore. But that just indicated that there was landmarks, but that was only for the final 5 kilometres. So they've got a map. What is that map? Well, the simplest explanation is the fact that it's a series of gradients. And these gradients have to always be available because navigation happens at all times of the year, all times of day, and all through, it doesn't matter what the weather is. There's got to be very minimal seasonal variation and daily variation. If things vary too much, then the animal wouldn't be able to interpret where they were. It's got to vary predictably and systematically, because when you take an animal to a location it's never been, like I did, the only way for it to fly back is if it can actually read the local information, compare that to the home information, and work out which direction it should head in. And finally, the animal has to have sufficient sensory resolution to detect that gradient. And we think here at Auckland Uni that it is the magnetic field, because it's the only thing that's been proposed so far that fits these 4 requirements. And if you look in the literature, you can actually see a lot of evidence suggesting that birds and other animals are using magnetic fields for a map. One of them comes from vanishing bearings from large groups of pigeons released on anomalies, magnetic anomalies. And that's the little white graph you can see there. So running along the x-axis, that's the one at the bottom, you can see the magnetic intensity increasing for an anomaly. And then running up the other side is the mean length of the vector, which is a way of measuring the degree of scatter. So if you've got a measurement of 1, everyone's heading in the same direction of one line. If you've got a direct mean vector length of 0.2, they're all heading in different directions. There's a large degree of scatter. They've all got a different idea of where they should go. And you can see there's a very strong negative correlation between those two variables. And as we know that celestial cues are used as the dominant cue, the best explanation is that they're using a magnetic map. The second one is that pigeons released around Auckland area by one of my lab mates have been shown to fly along magnetic intensity lines rather than in the home direction. So you can see 2 release sites here, and there's a solid yellow line running down, pointing towards the home direction. And then there's also several pigeon tracks in blue, red, and yellow, and white. And then there's the very thin lines. Those are the lines of equal magnetic intensity, sort of like topography. And you can see that the pigeons are actually flying along those lines. They're not flying in the home direction. So, how would you determine where you are? If you look at this is the Earth's magnetic field across the entire globe, and it's very easy to see how you would have a latitudinal magnetic cue, because the magnetic field is weakest at the equator and strongest at the poles, and it varies systematically. So since birds fly from up and down North America and up and down Europe and not between, it doesn't matter that at one latitude you've got a different magnetic strength than at another latitude, because they don't actually fly between those two locations. So that's very easy to understand and see how that could be used locally for a bird or a bat to determine whether they've been displaced north or south of where they want to go. But what about the longitudinal coordinate? This is one that's confused people for a very long time. Well, that was hiding in the literature as well. A paper published by Windsor in 1975 managed to do a very clever experiment where they released a whole lot of pigeons all around in the systematic releases around the loft. And what you're looking at here is symbols representing information from the release data. So each release site is pointing towards the center of the graph, and that's where the loft is. And there's a second bar going out to the right or the left, indicating the mean direction that the pigeons disappeared in. And the filled symbols show right-handed errors, and the clear symbols show left-handed errors. And you can quite clearly see that these are not randomly distributed. One set's on one side of the picture, the other set is on the other. And if you look at the lines of equal magnetic intensity, which are the 2 red lines, the slope between those lines, which is the blue line, neatly divides those observations into the 2 groups. There's a little bit of biological variation there, but that's the joys of biology. So it looks like it's that slope that could be our second coordinate. And so when we go back to our global map and you look at New York there, you can see that little bar that the slope's facing in one direction on the west coast and it's facing in the opposite direction on the east coast. And you can see how it varies systematically longitudinally. A similar pattern was found in Frankfurt when people repeated his experiment. So it looks like we've got our longitudinal method. So did I find any evidence like that for long-tail bats? Well, I found one track where the bat didn't go feed straight away or didn't go hang up and sleep in a tree, where the bat flew north and then south and then east, due west. And I got these measurements using radio telemetry. So you can see each of the fixes on a 20-second interval. And the red lines are the lines of equal magnetic intensity for the area. The problem is this is using radio telemetry. And radio telemetry is not very accurate. So I'm not very confident that we could use this evidence to show for certain that bats using the Earth's magnetic field. But it was a very encouraging first result, which is why I continued on and did a PhD after my master's to demonstrate that bats are actually homing using the Earth's magnetic field. And for this, I have to travel to South Africa because our bats are too small to carry GPS units. I needed a fruit bat. And there's a colony of Egyptian fruit bats living at the top of Table Mountain here, 800 metres up from the car park. I've climbed this mountain many, many times. I've lost count. And I was hosted by the University of Cape Town, David Jacobs' lab. So we also have a magnetic map for the area. It's pretty coarse, but it did allow us to pick a couple of sites. And you can see the cave there is a little hexagon, and then the two release sites as triangles. So the procedure was go up the mountain, catch a whole lot of bats that were big enough, bring them back to the student accommodation where I was staying, and attach the units to their back. And I did this so they'd actually get used to having something on their back, since these are wild bats. Then I pitched a tent in the lounge and kept the bats in there with some food and water. Shh, don't tell student accommodation. In the morning, I would then take the bats out to the release site and give them another feed of pure fruit juice mixed with honey, which they really loved, and that you can see the little guy there lapping it up. And then I'd let them go with the tag on their back, and they'd fly back to the mountain. This is a really good photo of the bat in flight. Terrible photo of me. I'm always concentrating really hard when I do this, so I forget to also suck it in and smile. There you go. That's much better. So what did I find? Well, those bats homed as well, and they homed back really quickly. You can see the 5 thick line tracks in black. Those are the bat tracks, and they went from the release site back to the cave on the same night. The thinner line tracks are homing pigeons that I released at the same location during the day, and their loft was in the same direction as the cave. You can see where they cut off a little bit earlier. One thing to note is that the homing pigeons weren't as good as the bats at homing, which was kind of interesting since the homing pigeons could fly around during the day, and also 3 of the bats that I released at this site were released in heavy fog, so they didn't have any visual cues either. So it suggests that the bats are better homers. When we zoom in really close, we can see a lot of that stepping behaviour that we saw with the bats, with the birds here in Auckland. So it suggests that the bats are using the same or a similar method of navigating. So that's quite encouraging. Maybe bats are actually using this magnetic field. I'm currently analysing this data and am having to develop my own statistical methods because no one's got anything that would do this yet. So stay tuned. So what are the conclusions and recommendations from all of this? Well, first of all, bats can home, and the mechanism does look like it's magnetic. We'll just do a little bit more stats on it, and then we can say for certain. But this means that we can be very confident that any hard release of adult bats will fail, because that's essentially what a homing experiment is. It's a hard release translocation. The only difference is what you'd consider to be success. Some people have suggested that we should just catch young of the year and translocate sub-adults. I'm not overly convinced about this because we don't know when they imprint on their roost. And if we are going to try that, we should do it from very close locations so the young can get back if they try to make it. So I think the best thing we should do is this procedure. So this is what we're having a meeting in November where I get to bring my navigation expertise, and a whole lot of other bat experts will be there bringing all their particular areas of expertise. There's zookeepers, for instance, coming from Auckland Zoo to teach us and design how we are going to actually hold the bats and look after them. But I'm focusing mainly on the release protocol, and this is the protocol that I want to use. I would like to take a whole lot of pregnant female bats and then take them to the location that we're going to be trying to translocate them to and have them give birth there. That means we're going to have to build our own facilities to hold them there rather than take them to a third location. Once the young are flying around the enclosures and they're happy and all that, and they're no longer breastfeeding from the adults, we're going to release the adults and see if that's enough, the soft release is enough to convince them to stay on the island. Maybe giving birth is enough for them to reset where they think home is. Now, if they all home back to the source population, well, that doesn't matter then. We're fine. We can seed the population with the remaining juveniles, and we just know that we can never take adult bats. No matter how long you hold them, they're still going to stay in the same place. But if the adults stay, maybe we've reset the homing mechanism. Maybe where they're considered to be home. Maybe we've actually cracked it. But we shouldn't be too rush into it too much, because it could be that mum's hanging around waiting for junior to come out. Observations from people who have worked with bats, especially from someone who actually trained me, suggest that New Zealand bats are really good mothers, which isn't surprising. One of my trainers, she found a baby bat in the bottom of a harp trap, and there was a mother bat, or as it turned out, flying around squeaking. And she took the baby bat out and held out her hand in the air, and the mother bat flew down, landed on her hand, tucked the baby up, and then flew off. So clearly there's a very strong bond there, and mothers will come in come in close to people to get to their babies. And so maybe that's what the adults would be doing. So we need to release a couple of juveniles with transmitters that we know who the mothers are, and see whether or not they disappear, because they could then fly back to where we caught them from, and junior could follow mum. So release a few little juveniles just to see what's going on. If the adults stay, then awesome, we've managed to completely translocate bats. If not, if they follow mum, well then we just hold on to them for a lot longer. We wait for the mothers to give up, and then we release the juveniles. So this is a surefire way of overcoming their navigation. And I learned all this stuff from doing my master's and PhD. So the next question is, okay, where are we going to take them from and where are we going to take them to? Well, this is some of the other stuff we'll be talking about. But what we do know is that the source population must be very healthy, since we're trying to develop a successful bat protocol. So this means we're going to be targeting a population with a lot of bats where they're already at carrying capacity, and us taking some bats from there won't do anything wrong. It won't affect them in any way. And where we're going to take them to, it's got to be a site with large trees, because bats need big old growth forest for their maternity roosts. And because predators are such a big problem, it has to be a predator-free location. So it could be somewhere like Rangitoto or Tiritiri Mātangi, or any of the other offshore islands that have no predators on them and also no bats, but big trees. But we could also take them to mainland sites if there is a community group or an area that has got a predator-free or strong predator control. So somewhere like Tawharanui, Ark in the Park, that sort of thing. Essentially anywhere that actually meets any of these criteria will be considered. Now, I'd like to thank everyone at Auckland University for teaching me and training me. I love the University of Cape Town in South Africa, the David Jacobs Lab for all their help, the supporters of Tiritiri Mātangi Island who funded me for my MSc, Colin O'Donnell and his crew from the Eglinton Valley and the Department of Conservation for training me on how to work bats, and of course my three favourite field helpers, Rebecca Hamner, Stephen Greff and Greg Matuiti, who helped me in the field. And thank to all of you for voting and sharing to get this project some of the initial seed funding that we need to get going. Look forward to seeing the result on Tuesday.